Hello everyone, welcome back to Mahi's Life Science channel. In this video, we will discuss about the multiplication of bacteriophage as well as their lytic and lysogenic cycle. Multiplication of bacteriophage. Bacteriophages are capable of multiplying only within the bacteria. They remain inactive outside the bacterial cell and they are host specific. For example, T4 bacteriophage infect and multiplies only inside the bacterium E. coli. And the multiplication of phage take place in the following steps such as attachment of phage on bacterium, injection of phage DNA into the bacterium, disruption of bacterial metabolism, assembly of phage particle and lysis of bacterium. First one is attachment of phage and bacterium. When the phage and the bacterial cell come close together by random collision. After that, the phage attaches itself to the surface of the bacterial cell wall. The tail fiber selects specific site on the bacterial cell wall and attach with it. Then the tail fiber bend to keep the end plate in contact with the bacterial cell wall. Second one is ingestion of phage DNA into the bacterium. The tail secrete the enzyme lysozyme which dissolve the bacterial cell wall to make a hole. The contractile sheet contract and this force the tail core into the bacterial cell through the hole. The phage DNA is then injected into the bacterium through the tail core. The protein coat does not enter in and it remains outside the bacterium. And the third one is disruption of bacterial metabolism. The phage DNA disturb the bacterial metabolism and synthesize phage DNA and proteins. The synthesis of normal bacterial proteins stop. Meantime, the phage DNA synthesize phage protein using the host protein synthetic machinery. The viral DNA produces mRNA which later form the phage proteins. The bacterial DNA is degraded into free nucleotide by the action of viral deoxyribonuclease enzyme. Therefore, the synthesis of bacterial DNA stop. At the same time, the phage DNA replicate by using the free nucleotide and produce many copies of phage DNA. This biochemical reaction is infected bacterium lasts for 4 to 10 minutes after the entry of phage DNA. Fourth one is assembly of phage particle. The phage protein forms subunit of capsid, tail sheath, tail fiber and end plate. The subunit form head. Each phage DNA copy then enter the head and become tightly packed. The tail sheath and the end plate join together to form a tail. This tail is then attaches to the head. Thereafter, the tail fiber join with the end plate to form a new phage particle. Thus, about 150 to 300 phages are produced in a single bacterium. The duration between the ingestion of viral DNA and the formation of the first phage is called eclipse period. It takes place between 10 and 20 minutes after the infection. And the fifth one is lysis of bacterium. As many phages have been produced in a bacterium, the bacterial cell wall ruptures suddenly. The new phage particles are released free. It takes place in 20 minutes after infection. Life cycle of phages. Bacteriophages exhibit two types of life cycle. They are lytic cycle, which is also called as a virulent cycle, and lysogenic cycle, which is also called as a temperate cycle. First, we will discuss about the lytic cycle. All the T series phages exhibit lytic cycle. These virus infect bacteria and on completion of the life cycle, they cause the lysis or rupture of the bacterium. Hence, the cycle is called lytic cycle. Lytic cycle involves the following steps such as attachment, penetration, replication, maturation and release. First one is infection take place by random collision between the phage and the bacterium. After that, the tail fiber selects specific site on the surface of the bacterium and these spikes anchor firmly. A hole is made in the cell wall of bacterium by the enzyme lysozyme secreted by the tail. The sheath contract and this causes the tail core to penetrate the cell wall. The DNA of the head is discharged into the cell through the tail core. The protein coat does not enter in and it remains outside the cell only. The DNA of the phage take over the protein synthesis machinery of the bacterium. The host DNA is degraded by the viral deoxyribonuclease. Viral DNA is transcribed into mRNA, and the mRNA translate protein which are utilized for the synthesis of viral proteins. Viral DNA also replicate to produce hundreds of copies. Each DNA copy is enclosed by a protein coat producing new virus. Soon the bacterial cell wall rupture and the new viral particles are released. A single phage can produce about 200 phages in 20 minutes after infection. Lysogenic cycle. All the lambda series bacteriophages exhibit lysogenic cycle. In lysogenic cycle, the bacterium is not lysed. As in the lytic cycle, the viral DNA enters the bacterial cell, but the viral DNA does not take over the protein synthesis machinery of the host. Instead, it gets integrated with the bacterial chromosome. At this stage, the virus is called prophage. The bacterium with the prophage is called lysogenic and it is resistant to other phages. The viral DNA replicate along with the bacterial chromosome. Rarely, the prophage DNA can disintegrate and enters into the lytic cycle. Thank you for watching this video. Like, share and comment. Subscribe for more videos.